Welcome back, folks. This is our kind of last content section before we do our technique for this chapter. And so in this section, we're going to look at the nucleus, not in too much detail, but we'll talk a little bit about nuclear structure and some of the challenges presented to the cell by actual virtue of having a nucleus. So as you know, the nucleus is bounded by a structure called the nuclear envelope. It's got an inner membrane and an outer membrane, and those two delineate a space called the perinuclear space. And the outer membrane is continuous with the endoplasmic reticulum, and it contains a number of different types of proteins that we're not going to get into the specifics of, that bind two elements in the cytoskeleton, the actin, microfilaments, and a variety of different types of intermediate filaments. In some cases, there are also tubular invaginations of the nuclear envelope into the nucleoplasm, which is the material contained within the nuclear envelope. Also on the surface of the nuclear envelope, we find ribosomes involved in translation, and we find structures called nuclear pores, and we'll look at those in detail in a little bit. Those are specialized channels that allow material to move in and out of the nucleus. And they form where the inner and outer membranes fuse and provide this direct contact between the cytosol and the nucleoplasm, the inside of the nucleus. And those pores are lined with a set of proteins which collectively form what is called the nuclear pore complex. So that's a little bit about the structure of the nucleus, a couple of new pieces of information for you. Let's take a quick look at this nuclear pore complex. So there's an electron micrograph over here, and you can see these pores are relatively dense on the surface of the nucleus, and they look very, very symmetrical and somewhat complicated in structure, having a number of subunits. So if we look at these in an artistic rendition, we see that there are a number of these proteins, and they're called nucleoporins, and there is a structure called a basket, and then there is a central transporter which is going to move material into and out of the nucleoplasm. So that's the overall structure. Let's look at the problem that we've got to overcome. So if you've got a nucleus and you've got processes occurring in the nucleus, then you often have to move the products of those processes into the cytosol. And by virtue of the fact that you have processes in the nucleus, you often have to move proteins and other molecules into the nucleus. So there are many enzymes and proteins that are needed in the nucleus that have to be imported from the cytoplasm, and RNA that has to be translated, mRNA, has to be exported from the nucleus. And in addition to mediating this transport of various proteins, for example those needed in transcription, and the export of RNAs and rRNAs and tRNAs involved in translation, the nuclear pore complex also is involved in the movement of small molecules, small proteins and ions. And so if you are a particle less than about 10 nanometers in diameter, you can pass through the pores at a rate which is proportional to your size. So if you're smaller, you're going to move very freely. If you're up at the 10 nanometer limit, you're going to diffuse through much more slowly. Beyond that 10 nanometer size limit, proteins have to be actively transported across the membrane through the nuclear pore complex, and this requires that the protein to be transported has a sequence of amino acid called a nuclear localization signal, and that signal enables the protein to be recognized by the nuclear pore complex and transported across the membrane. And the membrane transport here works in both directions, into the nucleus and out of the nucleus. So we've got to look at how we can get things like ribosomal proteins out of the nucleus and into the cytosol, so the translation can occur. So a nuclear localization signal is anywhere from 8 to 30 or so amino acids long. It often contains proline and the basic amino acids lysine and arginine. And And so if we have a nuclear localization signal such as this one attached to a protein, then the protein will go into the nucleus. So what we've done is added this tag to a protein which is normally cytoplasmic, and that drags that protein into the nuclei of cells. And all we're doing here are looking at the nuclei of the cells. If you mutate the nuclear localization signal so that we change this second lysine to a threonine, is what we see is that the protein then is actively excluded from the nucleus. So that shows you the importance of the sequence of amino acids in the nuclear localization signal. So how does this nuclear localization signal work? So, multi-step process, looks kind of complicated, but we can look at it in terms of 3 or 4 main steps. So the first thing that's going to happen is that a cytoplasmic protein down here with a nuclear localization signal is recognized by a receptor called an importin protein, and that importin binds to the nuclear localization signal of the protein that's going to be imported, and then the importin protein to be moved complex moves to the nuclear pore complex, and so the importin associates with the nuclear pore complex, and the whole thing is then transported through the nucleus, through the nuclear pore and into the nucleoplasm. Once inside the nucleoplasm, the importin and the protein that's been imported associates with a molecule, a protein called RAN, and RAN is a GTPase, and so it has a molecular GTP bound, and so RAN is responsible for dissociating the importin from the cargo, and so here's our cargo left inside the nucleoplasm. RAN GTP then associates with the importin, and that sends the whole complex back out through the nuclear pore complex, and then RAN GTP hydrolyzes the GTP to GDP. That releases the importin, and the importin can be released. And so this whole process is dependent on this protein called RAN. That's import, export happens in a very similar manner. RNA molecules are exported by adapter proteins that bind to the RNA, and those proteins contain nuclear export signals, and they target the proteins and the bound RNAs for export. So you think about the number of RNAs that are made in the nucleus. tRNA is being transcribed in there, ribosomal RNA is being transcribed in there, messenger RNA is being transcribed in there. They all have to be exported for the purposes of translation. So that's how we get the proteins in, and how we get the importin back out, and how we get mRNAs and tRNAs primarily back out of the nucleus. And then there's a whole cycle for getting the RAN GDP back into the nucleus so it can be involved in this part of the transport process. So enough about nuclear transport. Let's just look at the architecture of the nucleus and the chromosomes in the nucleus. Now, most of the time, the cell is in interphase. If a cell takes, let's say, 24 hours to divide, the events of mitosis and cytokinesis may only be 1 to 2 hours. So most of the time, the cell has its chromatin fibers being extended and dispersed throughout the nucleus in an uncondensed format. Now, you would I can imagine how people just easily think that it's just all the chromosomes are just kind of twisted around one another and just dispersed randomly through the nucleus. But in fact, each of the interphase chromosomes occupies a discrete region called a chromosome territory in the nucleus. And so it's what you can use as a technique called in situ hybridization, and this is largely what we're going to look at when we look at our technique in a moment. We can use a piece of DNA, a single-stranded piece of DNA, which is fluorescent, and we can cause that to hybridize with a complementary sequence of DNA. So if we have a sequence of DNA which is short and complementary to chromosome 7, if we treat the nuclei in some cells such that the chromosomes separate slightly, and we introduce the probe, the fluorescent probe will bind to chromosome 7, and then when we renature the DNA, what we'll have is fluorescence of the probe, and that will tell us where in the cell chromosome 7 is. So here we can see there are 2 discrete regions where we see chromosome 7 localized, and here we see another probe that's showing us the 2 copies of chromosome 8. You can see they occupy discrete territories, and the whole genome has been mapped in this way, so we can look to see where each of the unique chromosomes exists in its own territory in the nucleus. And so we'll take a look at a technique called fluorescent in situ hybridization in a moment with our last part of this section, to see how we can actually locate pieces of chromosomes and whole chromosomes in cells. So we'll do that in a moment. The last thing to talk about here is a structure called the nucleolus. And you'll often see this in textbooks. It's often some kind of discrete structure that's shown inside the nucleus. It's not membrane bound, so it's not a membrane bound compartment within the membrane bound compartment of the nucleus. It is just a very dense region of the nucleus, and it's so dense you can actually see it under light microscopes. So this is the place in the nucleus where ribosomal subunits are assembled. So this is where the cell actually puts ribosomal subunits, the small and the large subunits together, and so they have to be exported out of the nucleus through the nuclear pore complexes before they can be used in translation. And they're kept separate. They don't come together until you actually initiate translation. And so within the nucleolus, we also have these structures called fibrils. And those are regions of DNA that are being transcribed into ribosomal RNA. So that's regions of the genome which are highly active in terms of transcription of ribosomal RNA. And then there are granules, and those are structures where RNA molecules are being packaged into things like ribosomal subunits. So the RNAs are being complexed with proteins. Sometimes that will be for export, sometimes it will be into ribosomal structures. So that's a little bit about the nucleolus. We'll wrap it up there. When we come back, we'll talk about this cool technique called fish. So we'll go fishing. I'll speak to you soon.